Hello everyone, I'm very pleased to share the practical issues and the strategies of biological product technology transfer. About the technology transfer, I will focus on these 4 points: MSAT introduction, technology transfer strategies, practical elements of the technology transfer, and some case study. At the end, I want to introduce you the Genscript Biomanufacture Center. Firstly, I will talk about the MSAT, who conduct the technology transfer. The goal of MSAT is transferring a process feasible, compliant, and economical from PD to GMP production. This cycle shows all responsibilities of MSAT. To transfer a project successful, MSAT should take part in the project as early as possible, and MSAT must make sure that the plan designed by PD is feasible to scale up. Then, based on the data from PD, MSAT should conduct a risk assessment to preliminarily determine the CPP and analyze the gaps between PD and GMP. After this, measures to reduce these risks and the gaps must be made. Then MSAT should confirm the scale-up strategies and make the sample plan. Then, based on which, process description and batch record should be drawn up to instruct manufacturer. During the manufacture, technical investment of deviations happen in production, and plan the corresponding CAPAs is also the responsibility of MSAT. After enough production batches are finished, MSAT should build the scaled-down models, design the PC protocols by DOE, and instruct PD to conduct the protocols. After data analysis, design space should be determined, and based on which process validation can be conducted. As this slide shows, technology transfer can occur throughout the life cycle of a product, from preclinical development to phase 2 production. The main work of MSAT is scale up and process optimization. Then PC work should be conducted between phase 2 and phase 3 production. After design space was determined, process validation can be carried out. And nowadays, the concept of continuous process validation is more and more important. So MSAT should also monitor the commercial production continuously. As defined in ICH Q10, the aim of technology transfer is to achieve the commercial product by transferring the product and process knowledge between PD and the production within or between two sites. For example, scale up or scale down will happen between PD and manufacturer, and the production place change is also a very common situation in manufacture. Technology transfer will be easier to carry out when it happens in one company, since different sites in the same company share similar process and management, and the gap between them are small. But it's hard when a process is transferred from one production line to another. Although both production sites have mature systems, they both have abundant audition system, mature quality system, and enough skilled staff. They can't share the research experience, and the gaps of materials, equipment, and facility between them may be large. So usually, a research group should be included in the tech transfer team. A technology transfer committee is necessary. Since the management in transfer process is a challenge for most companies, The committee is a management team composed by department heads. They make decisions, manage the budget, timeline, and major changes. Then project manager and the head of each function can form a technology transfer group to conduct and push the project, solve problems in transfer process. Lastly, the implementation of technology transfer needs many function groups to take part in, such as PD, manufacturer, AD, QC, QA, and I. What's more, the engineering, finance, supply chain, and EHS also have a very important role in technology transfer. The technology transfer is more than a process. It should be taken as a project, and keeping the product quality is a key point in tech transfer. In the transfer workflow, the tech transfer process should be set as a project firstly, and the responsible people and the groups should be assigned. Then standing unit starts transferring related documents, and the tech transfer group review these documents and conduct a risk assessment. Once risks are identified, protocols to reduce these risks should be planned. The MSAT can draw up technology transfer protocol. After this, the tech transfer can be implemented, and it contains 3 parts. There are manufacture process transfer, analytical methods transfer, and clean process transfer. This transfer process can be firstly implemented in small scale to reduce risks. If the process can be reproduced in small scale, we can start the engineering run and the GMP run in large scale. Then a conclusion can be presented in the technology transfer report if the product quantities are comparable. And at this time, the technology transfer is finished. Here we conclude some key points to ensure the technology transfer successful. The first point is document or information transfer. It is composed by analytical methods, quality attributes, production process, and validation methods. Systematic document transfer can help receiving unit understand the process, analyzing gaps, and as a result, increasing transfer successful rate. The second point is gap analysis. We can analyze gaps from people, equipment, materials, methods, environment, and testing 6 aspects. After the gap analysis, the third key point, risk assessment, should be conducted. As we can see, risk analysis is throughout the whole process of the technology transfer, especially in CQA and CPP evaluation and process validation. Then I will give some examples to show how gap analysis and the risk assessment are conducted in a tech transfer. This slide gives some gaps in the project transfer. The gaps cover upstream, downstream, and materials. For example, the bioreactor changes from applicant to solver when scale up. The bioreactors have different geometric design and exposures. It can be treated as a high risk because the CQA of product may be affected. So when scale up, we need to consider the gas strategy and the mass transfer. For chromatography system, different light paths and flow rate should be considered, and this can be treated as medium risk. since they only affect the recovery rate. Many people have different kinds of ultrafiltration system, but they have the same principle, and the flow rate is the only key point should be considered. So we can take it as a low risk. When materials change in technology transfer, we should evaluate the function of the material and assess the risk level based on its use. In Probio, we will take many steps to reduce these risks. For example, we compare parameters and build models for different bioreactors to make scale-up or bioreactor change successful. In cell culture, cell density is an important factor, and the cell count equipment may have great effects on cell density. To eliminate these effects, we compare the results of two cell count equipment and find the relationship between them. As we mentioned above, different chromatography systems have different UV absorbance. So we fit some model to predict the UV absorbance. And thanks to these models, we can determine the connection start time and end point. When scale change in technology transfer, two kinds of parameters should be considered. First kind is scale-independent parameters. For example, in cell culture, there are pH, temperature, and feeding strategy. In column packing, there are column height, efficiency, and symmetry. In chromatography, there are retention time, loading capacity, pH, conductivity, and the connection parameters. In filtration, there are memory type, capacity, and processing time. For these parameters, we just need to identify them and keep them constant. However, when scale changes in cell culture, we should modify the storing speed, spike strategy, and the catch volume. In downstream, the column volume, loading volume, memory error, and the flow rate also should be modified based on different process. Since we complete the points mentioned above, Probio have complete many successful technology transfer. Here I will list some examples of them. Case 1 is the biosimilar. From the pictures, we can find that cell growth, tide, and PTM are all within the double standard deviations. From this table, we can conclude the quality of products produced in different scales and in different sites are comparable. All of these indicate the technology transfer is successful. Case 2 is a monoclonal antibody, which transferred to a new factory. We can see from the pictures, the cell growth, metabolism, titer, and the product quality are constant in different batches, and they are comparable with small scale. In downstream, the recovery rate of each unit are similar, and from the SEC results and the HCP residue, we think the purity are also comparable. Case 3 is the tech transfer from Probio to client. After process development, Genscript provides comprehensive and refined technology transfer to the client, helping the client finish technology transfer in 1 month. Now the client has finished 3 batches of GMP manufacture. To conduct a successful technology transfer, we think that these 4 key points are necessary. Firstly, a good work relationship. that is a fair, trusted, and respected relationship. Secondly, sending units should be willing to share the necessary documents, experience, and materials. Thirdly, a good team works with the same target and can cooperate with each other to solve problems quickly. Lastly, communicate the details of the project at early stage and make some strategies to reduce risk. That's all the technology transfer strategies I want to share. And at the end of this presentation, I would introduce our company, Genscript Probio. At Probio, we are all about innovation through collaboration, and we will proactively serve our clients, treat you professionally, and make sure our process is the most efficient one. Our mission of Genscript Probio is committed to helping customers to shorten the timeline for development of biological drugs from discovery to commercialization, significantly lowering the R&D cost and building a healthier future. In Probio, we have cutting-edge platforms of drug discovery, both in monoclonal antibody and bispecific antibody. We can also provide the total solution covering the product life cycle, from preclinical development to commercial manufacture. This is our track record. We have more than 30 CMC and 3 R&D approvals in the recent 2 years. What's more, we have achieved many achievements in the last year, such as new GMP manufacturing center put into production, having 14 CMC or CMO orders, and 4 orders are from overseas. In 2020, we complete 20 GMP batches and 9 custom auditions with no critical and major matters. We only take about 8 months from DNA to the first 2,000 liter batch. And in perfusion, we also get a good result. We can have a volume production more than 6 grams per liter per day. This is our factory. The first building presented is our R&D building. Our in vitro pharmacology discovery platform and the CMC platform are all in this building. The second building is our new GMP manufacture center, which has the capacity of 2,200 liters. Genscript Probio is welcome for your visiting and cooperation. Thank you for your listening.